my channel. So I have a YouTube channel where I upload all my recording for my lecture in that YouTube. So later on, I will give you the link. Okay. So all right. So basically, today's lecture just a short recap of our last lecture. So last lecture we have generally looked on the overview of the different types of macromolecules. Okay, so we have four macromolecules. We have protein, carbohydrates, lipid, which is lipid, and then the last one is nucleic acids. Okay, so there are four macromolecules. So apart from lipid, protein, carbohydrates, and nucleic acids, they all have monomers. Okay, they all have monomers, and these monomers are joined together to form a polymer. Okay, a polymer. So that's why in this slide it says here synthesis and breakdown of polymers, referring specifically to the formation of polymers for protein, carbohydrates, and also nucleic acids. Okay, so how do you attach monomers together to form a polymer? Okay, so in order for the reaction to occur, to attach together the monomers to form a polymer, the reaction involved is dehydration. Okay, dehydration reaction. So if you look at the term dehydration, the reaction involves the removal of water molecule. Okay, involves the removal of water molecule in order to break in order to break bonds. Okay, in order to break covalent bonds between the atoms of that polymer. Okay, to break the bond between basically the monomers. All right, so that is dehydration. uh, uh, to break bond, okay, through the release of water molecule. Okay, dehydration. So dehydration removes a water molecule forming an sorry, saya terbalik sorry. Dehydration is to synthesize polymer. Dehydration is to synthesize polymer. So it means that you attach monomers together, you attach monomers together to form a polymer. Okay, so dehydration. So in order for the reaction to occur, to attach one monomer to another monomer to another monomer, so this involves the removal of water molecule from the monomer. Okay, saya bagi contoh kat sini. We have, for example, an amino acid. Okay, the structure is amino acids. I've drawn this before in the class. So it has a hydrogen, a side chain, and also an amino group. Okay, so and then at the end here it has the hydroxyl group. So bukan hydroxyl, carboxyl group. Okay, OH. Okay, so this is one amino acid. So another amino acid, for example, I draw using purple ink. So it has nitrogen, hydrogen, and then here is the carbon, side chain, hydrogen, and also the carboxyl group. Okay, so this is one monomer of amino acid. So this is another monomer of amino acid. The reaction is dehydration to attach the monomer together to form a polymer. So in this case, because it is dehydration reaction, a water molecule will be removed. So in this case, if you look at the reaction, the molecule properly, okay, so what are the reaction involves? Here it removes a hydroxy group from the carboxy group, the OH from the carboxy group, and then also a hydrogen atom from the amino group of another monomer. So this, you remove OH, you remove hydrogen atom, it becomes a water molecule. Okay, so a water molecule. So once you remove the OH and also hydrogen atom to form a water molecule, so you can now remove this ion, the atoms, and then you will form a covalent bond. Okay, you form a covalent bond between the monomers. So this is a type of covalent bond, because this it involves the attachment of one amino acid to another amino acid. So this is a peptide bond. Same goes to other macromolecules. So if it involves the attachment between glucose to another glucose to form maltose, or glucose to another glucose to another glucose to form starch, for example. So this involves the attachment of the monomers through the to the reaction. We call it as dehydration, removal of water molecule during that reaction. Okay, so the next one is you have a polymer, and then you want to separate the monomers, okay, from the polymer. So in order to separate the monomers from the polymer, you need to break bond. Okay, break bond. So break bond, and the reaction requires water molecule. So that's why the reaction next is hydrolysis. Hydrolysis, the reaction involve water molecule to break bond. Hydro is water, lysis is to break bond. Okay, so here as you see, we have a polymer, a polymer that consists of several monomers. So you want to break the bond between the monomers. To break the bond, you want to break the bond. So in order to break the bond, the reaction requires a water molecule for every bond that is broken between the monomers. Okay, so it requires a water molecule. Okay, so dekat sini as you can see, contohnya kita dah ada, let's say here, initially I've drawn this is a molecule, we call it as dipeptide. Okay, so if you add more amino acid, it forms a polypeptide. So basically, in order for you to break this peptide bond, you need to add water, and you need to add water. So hydrolysis is the opposite reaction of dehydration. Dehydration is the opposite reaction of hydrolysis. Okay, so samalah kan. So because it is hydrolysis, water molecule will be needed to add again the OH to be attached to this carbon, kan, OH, and then the hydrogen is attached to the amino group. So you break this bond and dapat baliklah amino acid, kan, the two amino acid will be separated. So the same concept applies to all the monomers in polymers. Okay, so tak kisahlah ada protein ke, karbohidrat ke, ataupun uh, nucleic acids. Okay, the attachment between amino acids, the attachment between the monosaccharides, the attachment of nucleotides, they are all attached through dehydration. They are all will be broken, the bonds, through the process of hydrolysis. Okay, so if you have question, you can ask question. Okay, so next, that is the introduction. Before we go deep into each of the macromolecules. So the first macromolecule we are going to look at is protein. Okay, protein. So protein, okay. So it says here protein consists of one or more polypeptides. Okay, one or more polypeptides. A protein molecule can consist of only one polypeptide chain. It can consist of between the interaction between several polypeptide chains. Okay, so the monomers for the protein are the amino acids. So there are 20 different amino acids. Okay, 20 different amino acids. Feature of each amino acid, it has here a carbon atom. Okay, we call it as a chiral carbon. Chiral carbon, can carbon at the center of that molecule. Okay, the alpha carbon here. And then we have attached to that carbon, there's another four different groups. Okay, four different groups. So the carbon will be attached to the amino group, attached to the carboxy group, attached to hydrogen atom, and attached to the side chain or the R group. Okay, so each one of you is required to remember on how to draw an amino acid. This is the basic structure of amino acid, of which you have to remember or know how to draw. Okay, so each amino acid has different side chain. So the side chain determines the 20 different types of amino acids. So the side chain will differ. So there are side chains that have, for example, hydroxy group. There are side chain that only has hydrogen atoms. There are side chain that has carbon and also hydrogen, the hydrocarbons. Okay, so there are side chain that also has amino group. Okay, so there's also side chain that has carboxyl group. Okay, so based on the side chain of that amino acid, it determines the property of the amino acid, and indirectly also determines the property of that protein. Okay, so the R group of amino acid can vary in structure, okay, in charge. So the side chain can have positive charge, negative charge, or neutral. So if the side chain has, for example, amino group, it will be positively charged. If the amino group has side chain that has carboxyl group, it will be negatively charged. If the side chain of the amino acid has hydrocarbons such as only hydrogen atom, or it has methyl group, for example. So this causes the amino acid to be neutral or non-polar. Okay, non-polar. So the R group of amino acid can vary in affinity with water and their reactivity with other molecules. Okay, so affinity of water. So for example, if the side chain has hydroxyl group, okay, it has hydroxyl group OH. So this allows the amino acid to have a polar property. Okay, polar property, polar. It can form hydrogen bonds with water molecule. Okay, can form hydrogen bond with water molecule. So for example lah, contohnya, tak apa, nanti we look at later the different structure of the 20 different amino acids. So there are 20 different amino acids which commonly found in proteins, each identified by the different R groups. Okay. So amino acids are grouped by the properties of the side chain. R groups can be polar if the side chain has hydroxyl, for example, or it has sulfhydryl, for example. Non-polar if it has consists of hydrocarbon, only carbon and also hydrogen. Acidic if it has carboxyl group, COO negative, and hence the amino acid will have a negative charge. And then basic if it has amino group, and hence the amino acid will be positively charged. Okay. So let's look at each type of side chains. Okay. So as you can see, this is the different types of amino acids. Okay. So in this slide, it shows you the amino acid that is hydrophobic. Hydrophobic amino acids. So this type of amino acid, they are hydrophobic because the side chains consist of hydrocarbons. As you can see, the yellow region here is the side chains. Okay, so if you observe at the structure of that of this amino acid, all this amino acid, the side chain only consists of carbon and also hydrogen. Okay, so for example, the simplest structure of amino acid here is glycine. Glycine, the side chain consists only of hydrogen atom. Okay, and as for alanine, it consists of methyl group. Valine, it consists of even more carbon and also hydrogen, hydrocarbons. Leucine, also hydrocarbon, isoleucine, proline, tryptophan, phenylalanine, and also methionine. So for this one, at your level, you are not required to memorize every single structure of this amino acid. You are not required to memorize every side chain. But the thing that you have to take note is that when a given structure of amino acid is given in a question, for example, you are able to identify, oh, this is a type of amino acid that is hydrophobic because it has hydrocarbons. Okay. Hydrophobic, it means that it will not attract to water molecule. Okay. Hydrophobic. So the next type is polar side chains. So if you look at this amino acid, the green region of the amino acid is the side chain. Okay, it is the side chain. As you can see, for polar amino acid that has polar side chain, it causes the molecule to be hydrophilic and attract to water molecule. Why? Because as you can see, the side chain has hydroxy group, for example, hydroxy group, or even here is sulfhydryl group. Okay, hydroxyl, another hydroxy carbonyl allows the amino acid to have a polar property, carbonyl, and also here the amino group also allows the molecule to be polar, can. And then also glutamine also has carbonyl group. Okay, with the presence of these different that's why earlier in this chapter we look at the subtopic functional group, so that you know, okay, if hydroxy it is polar, if it's carbonyl it is also polar molecule again. So polarity. So by having this type of functional group, it allows this amino acid to be polar, hydrophilic, will attract water molecule. Okay, so the next group kan, so tadi ini yang pertama, the first one. This is the second one. Okay, the second group of amino acid. And then the third one, yang ketiga ni dia terbahagi kepada dua. So electrically charged amino acid. Because the molecule of amino acid has charge, either positive or negative charge, it causes the molecule to be hydrophilic. It can attract water molecule. Okay, it can attract or interact with water molecule. So if you look at the side chain, for the amino acid that has negative charge, electrically charged amino acid, you will see that it has carboxyl group. Okay, carboxyl group. So amino acid that has carboxyl group, the carboxyl group will be ionized and becomes negatively charged. Kan? So carboxyl group asalnya apa? COOH kan. So bila ionized dia jadi COO negative. Kan? So because it has a negative charge, it can attract water molecule. Okay, so causing the amino acid to be hydrophilic. So this is acidic amino acid, that is the molecule have negative charge. And as for the basic amino acid, you look at the structure, you look at the side chain, it has positive charge due to the presence of amino group. Okay, due to the presence of amino group. Amino group initially, dia punya formula is NH2 kan. So when it is ionized, it becomes NH3 plus. Okay, NH3 plus. Dia macam ni. Okay. So the plus sign is actually the charge on the nitrogen. Yeah, nitrogen. Okay, so this is electrically charged amino acid, causing the amino acid to be hydrophilic. It can be acidic, negatively charged, or basic, positively charged. Okay. So again, you are not required to memorize the specific structure of every amino acid. All you have to know is that when a given structure of amino acid is asked during the exam or test or whatever, you have to you have to be able to identify the type of amino acid, whether it is polar, non-polar, acidic, or basic. Okay. Okay, so that is the the different groups of amino acid based on the side chain. So now we are going to look at the property of the amino acid. So amino acids are amphoteric molecules. So this term you have to know. You have to be able to spell it correctly, and you have to know what is amphoteric. Amphoteric is the ability of a molecule to be both acid and base. Okay, to be both acid and base. Because the name is amino acid, okay, it can be both base and acid. Both basic and also acidic. Okay. So that is amphoteric, can be both base and also acid. Okay, so that is amphoteric. So for amino acid, when it is dissolved in water, the amino acid will be ionized to form a Zwitter ion. Okay, so Zwitter ion is a molecule that has both positive and negative charge. Positive and negative charge. So the basic amino acid will be a proton acceptor, which will be ionized to NH3 plus. The carboxyl group will be ionized to form COO negative. So molecules with amphoteric properties can function as buffers. Buffers referring to specifically here is pH buffer lah. pH buffer, okay, pH buffer. To what is pH buffer? To resist any change in pH and try to maintain the pH. The presence of the amino acid, the protein inside certain aqueous solution uh, will will cause that solution to be maintained at certain pH. And so the pH of that solution will not change drastically, will not change drastically to be acidic, for example, or to be more too basic, for example. So yang ini saya draw sekejap in order for a more clear explanation, I suppose. Okay. Okay. So this is a much. So I have here a blank page. Okay. So now we are going to draw an amino acid. Kaitkan dengan the property of the amino acid being amphoteric, okay, amphoteric and also a pH, sorry, amphoteric and zwitter ion. Okay, so here is the basic structure of amino acid. So kita ada the carbon at the center, bonded to the hydrogen, the side chain, the carboxyl group, and also the amino group. Amino group. Okay. So ini dalam keadaan asal dia, tak belum ionized lagi. Okay. So if this amino acid were to be placed in aqueous solution, it will be ionized. Okay, it will be ionized. What happened during this process? Okay. So okay, kita tahu tadi amino acid is amphoteric. Okay. It can be both base and acid. Okay. Okay, base and also acid. Okay, acid. Okay, so ionize, the process of ionization. So in this case, what happened is it involves the OH here to be, the hydrogen ion to be atom to be released into the solution. Release, okay, positively charged. When the hydrogen atom of this carboxyl group is released, ionized, it may cause the pH to drop, the surrounding solution to be acidic. Can you increase the amount of hydrogen ion in the solution? Okay, so what happened is that this hydrogen ion, when it is released, okay, the molecule will become like this. It has a negative charge. Okay, the O, it releases the hydrogen ion into the solution, so the oxygen will be negatively charged because it loses a proton. It loses a proton and the oxygen becomes negatively charged. So and then hydrogen, the side chain, okay, amino, the amino group. Okay, the hydrogen ion here, it will be accepted by the amino group. It will be accepted by the amino group. So therefore, the amino group will accept the proton and become positively charged. Okay, so this is when this amino acid has both positive and negative charge. Again, it has both positive and also negative charge. So this is a zwitter ion that has both positive and negative charge. The amino group here, it becomes a proton acceptor. Okay, acceptor. The carboxyl group here, it becomes a proton donor. Okay, donor, it donates the proton to the amino group. Okay, so this is a zwitter ion, a molecule that has both positive and negative ion. So amino acid is amphoteric because it has the property of being acid and also base kan, acid and also base. So itu awak kena faham kan. So this is a zwitter ion, it has both positive and also negative charge. Okay, so amino group is a proton acceptor, the carboxyl group is the proton donor. Okay, so tadi kan saya lupa nak mention, okay, once the carboxyl group releases hydrogen ion into the surrounding solution, the pH will drop, becomes acidic. The pH of the surrounding become acidic. So this hydrogen ion will be accepted by the amino. So the pH will increase semulalah ke takat yang sebetulnya. So this is the function of amino acid of being a pH buffer. Okay, buffer. It resists changes in the pH of the surrounding solution, prevents the surrounding solution from becoming too acidic or too basic. Okay. Boleh faham? Okay. So thank you for responding.
Boleh.
Okay. Okay, so next, amino acid polymers. So amino acid are linked by peptide bond. One amino acid attached to another amino acid, attached to another amino acid. The linkage between this amino acid to form a polymer, we call it as peptide bond. So peptide bond is a type of covalent bond because it is the bond between atoms in the molecule. Polypeptide chain is considered as a molecule. Okay, so polypeptide bond is considered as covalent bond. It is a strong bond between the amino acid. So a polypeptide is a polymer of amino acids. Okay, so we have here already a dipeptide. Okay, a dipeptide. So you want to attach the third amino acid to the dipeptide to form a polymer. Okay, a polymer. So we know already the structure of amino acid, can? So for example here, the second amino acid here, it has a hydroxy group. Okay, so the amino acid that you are going to add, the third amino acid, will have hydrogen at the amino group. So the process involved here is what? Dehydration ataupun another term you can use is condensation. Okay, where the reaction involves the formation of bond and the release of water molecule. Okay? So OH will be removed from the carboxyl group, hydrogen will be removed from the amino group. They both will be removed and form a water molecule. Okay, so for each peptide bond form that attach amino acid together, for each bond forms, it releases a water molecule. So it means that if you need to add another amino acid, it releases the next it also releases a water molecule. You want to add another amino acid, again formation of the peptide bond also involve the release of a water molecule. So remember, for the formation of peptide bond at the carboxyl group, it releases the OH from the carboxyl, and for the amino group, it releases the hydrogen atom. Okay, so so this is the reaction, we call it as dehydration or condensation. Okay, so now you form a polymer. Okay, a polymer, in this case, the polymer consists of, in the diagram, consists of 3 amino acid. It can consist of many amino acid attached together actually. Okay, so for the structure of a polypeptide, if you look at the structure, the polypeptide molecule, the feature of a polypeptide molecule is that it has a backbone. Okay, a backbone. Okay, so the backbone is the same. It is the repeating unit of of these atoms. We have here nitrogen, carbon, carbon, nitrogen, carbon, carbon, nitrogen, carbon, carbon. Okay, so at the beginning of the polypeptide chain, at one end, we have here, we call it as the N-terminus. Okay, N-terminus, because at one end here it has the amino group free. Kat situ, kat hujung tu ada satu amino group free daripada amino acid yang paling hujung. Hujung lagi satu kan, the other end of that polymer, it has a carboxyl group. So this one we call it as the C-terminus. The N-terminus because it has amino acid. On the other end, it has the carboxyl, so we call it as the C terminus. Okay, amino end and the carboxyl end. Okay, so it is the same for every polypeptide chain. So it consists of N terminus, nitrogen, carbon, carbon, nitrogen, carbon, carbon, nitrogen, carbon, carbon. At the end, it has the carboxyl end. So what makes one protein differ to another protein? It is due to the side chains. Okay, side chains. So side chain, for example, the first amino acid here, it has this type of side chains. Okay, so it has carbon, hydrogen, and also here it has sulfhydryl initially, kan. So kat sini kalau kita tengok yang sebelum ni, kalau itu adalah kat mana dia duduk? Kat sini. So dia adalah tadi adalah non-polar amino acid because it has high amount of carbon and also hydrogen. Walaupun ada satu je sulfur because ratio dia dia ada lebih banyak carbon and also hydrogen that makes the amino acid to be hydrophobic. Okay. So kita ada hydrophobic amino acid, kita ada hydrophilic amino acids, polar. Here we also have polar amino acids, kan? So the different arrangement, okay, of amino acid along the polypeptide chain will determine the property of the proteins. It determines the property of the protein. If the amino acids of that protein have a lot of amino acid that has polar side chains, so it may cause the amino acid to be the protein to be a polar molecule, hydrophilic, that leads to the formation of maybe a globular protein, and protein that is able to be dissolved in water, for example. Okay. Okay, so this is a polypeptide chain. It has a backbone, repeating unit of nitrogen, carbon, carbon, nitrogen, carbon, carbon, and protruding from that backbone, you have the side chains. So these side chains will be involving in formation of interactions between the amino acid along the polypeptide chain. Later, we are going to look at what is the significance of having these different side chains. Okay. So, okay. So, classification of protein. So, protein can be classified based on levels of organization. How the amino acids are arranged. Not the amino acid, but the degree of tak apa, nanti kita tengok levels of organization. So levels of organization, it is we have 4 levels, okay, which is the primary structure, okay. We have also the secondary structure, tertiary structure, and quaternary structure. Okay, so levels of organization. All protein will have mesti di-startkan dengan primary structure. Okay, but not all protein will have quaternary structure, macam tu. Okay. So and then protein can also be classified based on its structure. Its structure. So structure can be for this one, the protein can be of globular protein. Okay, globular protein or fibrous protein. Fibrous protein. Globular proteins are protein that can dissolve in water. Okay. Fibrous protein are protein that cannot dissolve in water. Okay, based on its structure. And then composition. So composition, there are protein that is 100% made up of only amino acids. Okay, there are also proteins that have other component in that protein molecule. So for example, kita ada hemoglobin, kan? So hemoglobin, in the structure of hemoglobin, it has a hem group. Okay, inside that hem group, it has a ferrum. Okay, of iron ferrum. So the function of the ferrum in the hem group of hemoglobin is to bind to oxygen. Okay, so composition. So there are protein that consists of only amino acids. There are also protein that has other non-protein component that facilitate the function of that protein, facilitate the function of that protein. So the it may a protein can be activated. For example, it have if it has a cofactor embedded inside the protein structure, such as certain enzyme can. So that is composition. So protein that has other non-protein component, we call it as conjugated protein. Okay, conjugated protein, and we can also classify protein based on its function. Okay. Okay, so for four levels of organization in protein, primary structure is linear sequence of amino acids. Okay, so linear, kita alih lagi kejap dekat slide saya tak alih lagi, still on slide. Dia, kejap. Okay, dia dah alih. Alright. Okay, so four levels of organization in protein. So you classify protein based on levels of organization. So the first level is primary structure, which is basically involves amino acid attached to another amino acid attached to another amino acid through the formation of peptide bond. Okay, linear sequence of amino acids. That is the basic levels of organization. Nazan ni sekejap. Kita pause kejap. Okay, so that is the primary structure. And as for the secondary structure, results from hydrogen bonding involving the backbone. Okay, so kita tengok dulu definition dia, nanti kita akan tengok detail. So tertiary structure interaction among various side chains, which is the interaction between the R groups of amino acid along the polypeptide chain. And then number four, the fourth level is quaternary structure. So the interaction among multiple polypeptide chains. Okay. So for the first, second, and tertiary structure, you have to remember it only involves one polypeptide chain. Okay, one polypeptide chain. For the quaternary structure, this one it involves the interaction between two or more polypeptide chain that are interacting to form a functional protein. Okay, so for the first one is primary structure, simple linear sequence of amino acid joined by peptide bonds. So we have 20 different amino acids. So for different types of protein, they will have different arrangement, different sequence of the amino acid along the polypeptide chain. So enzyme A, enzyme B, enzyme C, hormone A, hormone B, hormone C, for example. So the sequence of the amino acid will be different, and hence it gives the property of that protein. Okay. So primary structure encodes genetic codes in DNA molecule determine the amino acid sequence of a polypeptide. So yang ini in order for you to understand this statement is basically in your cell there's the nucleus, inside the nucleus there's your DNA. Okay, your DNA is your genetic information. Consists of a sequence of bases. Okay, sequence of bases. So the sequence of bases in this DNA, okay, we call it as gene. Okay, different sequence of bases in the DNA code for the synthesis of certain protein. Kan? So based on this information in your DNA, which we call it as gene, from that it stores the information, and from that DNA, that information will be copied into a smaller version of molecule, which is RNA. So RNA is single stranded, and from that information, it will synthesize the protein specific protein. So this protein synthesis can be an enzyme, it can be a hormone, it can be other different types of protein that the cell requires. Okay. Okay. So second, the secondary structure segment of a polypeptide chain that coiled or folded lead to the protein's overall shape. Coils and folds are the results of hydrogen bonds between the carboxyl and amino groups of the polypeptide backbone. So there's the coiling of the polypeptide chain is we call it as alpha helix. The folded structure is we call it as beta pleated sheets. Folded. Okay. So some either the coil or folded. Okay. So the coiling and the folding of this polypeptide chain is due to hydrogen bonds. Okay, hydrogen bonds. Macam itulah, ini hydrogen bonds. Okay. So kalau tengok kat gambar ni, nampak dia kalau untuk secondary structure that involves pleated, dia macam nampak ada several polypeptide chain interacting kan, sebenarnya dia adalah satu polypeptide chain. Dia macam ni. Macam tu. One polypeptide chain only. Okay, one polypeptide chain. Okay, so let's look at the first one, which is the alpha helix. So the polypeptide chain is coiled. Okay, still one polypeptide chain only. So the polypeptide chain is coiled to form a helical structure. The helical shape is maintained by hydrogen bonds. Okay, hydrogen bonds. So you have to take note that for secondary structure to be maintained, it is due to hydrogen bonds. Okay, between oxygen of carboxyl and this is the carboxyl group. Carboxyl group, it has asalnya dia carboxyl lah, bila dalam keadaan ni orang nampak dia macam carbonyl kan. Tapi sebenarnya dia ada, sebelum tu dia adalah dia ada initially dia ada OH kan kat situ. Tapi sebab dia nak form bonds, so OH ni release tadi kan dalam proses dehydration. Okay. So between oxygen of carboxyl group of one amino acid and hydrogen of amino group of the fourth amino acid along the chain. So kita ada kan contohnya kat sini. 1, 2, 3, 4. Okay, so it forms hydrogen bond between every four amino acids, every four. Okay, 1, 2, 3, 4. So nanti dia akan form hydrogen bond. Macam tu lah. 1, 2, 3, 4. It forms hydrogen bond. So this maintains the helical structure of the amino of the polypeptide chain. Okay? So it is due to the interaction between oxygen and hydrogen along the backbone of the polypeptide chain. So this is the backbone lah, kan? Tadi kita tengok apa backbone? It consists of nitrogen, carbon-carbon, nitrogen, carbon-carbon, nitrogen, carbon-carbon. It has N terminus and C terminus at both ends. Okay. And as for the next structure is beta pleated sheets. Kita habiskan ni dulu sekejap ya. Beta pleated sheets, two or more segments, bukan two or more polypeptide chain, two or more segments of a single polypeptide chain that are arranged parallel to each other and held by hydrogen bonds. Okay, so hydrogen bonds hold neighboring strands in different region of a polypeptide chain. So the polypeptide chain become folded, has high resistance to stretching, and it is strong and also flexible. So again, for beta pleated sheets, it involves only a polypeptide chain to be folded. Okay, and the folding is held by hydrogen bonds between parallel region. Okay, between parallel segment. Okay, so kita sambung balik ya. Can I extend this session until we finish the subtopic of protein, please? Sikit lagi.
Boleh, madam.
Yeah. Okay. So sikit lagi je. Saya nak habiskan protein saja. Okay. So primary only involves the interaction between amino acids through the formation of peptide bond. Secondary is the coiling or folding of polypeptide chain in helical form or pleated sheets. So that is secondary due to hydrogen bonding. Saya nak tanya, kalau untuk secondary structure, what other bonds involved? Will it involve also peptide bond? Dia akan melibatkan juga peptide bond juga ke tak? Secondary structure mesti ada peptide bond. Okay. Secondary structure mesti ada peptide bond because it is the basic bond that holds the amino acid together. Okay. So primary ada peptide, secondary pun ada peptide, cuma nak hold the coiling structure and the pleated sheet is through hydrogen bonding. Okay. So the third structure is tertiary structure. Okay. Tertiary structure is the overall 3D shape of a polypeptide, again a polypeptide chain. Okay. Okay, a polypeptide chain that results from interaction between R groups of amino acids. So this interaction between the R groups include hydrogen bond, ionic bond, hydrophobic interaction, van der Waals interaction. You can also add here disulfide bridge. So these are all the interactions that hold a polypeptide chain to be folded to form a functional shape. Okay, so strong covalent bonds called disulfide bridge may reinforce the protein structure. So if you look at this diagram here, for example, yang purple ni adalah backbone lah. Tadi kan ada nitrogen carbon-carbon, nitrogen carbon-carbon. So protruding from that protruding from the backbone is the side chains. Okay, so let's say along the polypeptide chain here we have one amino acid, of which this the R group or the side chain, it has hydroxyl group. Okay, along the polypeptide chain there is another amino acid. Okay, that consists of only hydrocarbon, carbon and hydrogen. Along here, you have another amino acid that also has hydrocarbons. Along here, we have another amino acid where the side chain has carbonyl and also hydroxyl. Both are polar functional group. So they will interact to form hydrogen bonds. So asalnya polypeptide tu dia straight kan kalau primary structure. So bila dia jumpa kan one amino acid along that polypeptide chain, it also meets up with another amino acids that has polar side chain. It leads to the formation of hydrogen bonds. So this causes the protein to fold into a specific shape. And here you see that this amino acid and this amino acid has hydrocarbons. So hydrocarbons and another hydrocarbons, it forms an interaction which is hydrophobic interaction. Okay, hydrophobic interaction because the side chain consists only of carbon and also hydrogen. And another thing that you have to take note, for the amino acid that has hydrocarbons as side chains, they are non-polar kan, ataupun hydrophobic. You look at the structure of the protein, kan, hydrophobic, maksudnya apa? Dia tak suka air. It does not attract water molecule. So the position of this hydrophobic interaction will be isolated from the water. It will be maybe, for example, within the structure of the molecule that is away from being interacting with water molecule. Okay. So along the chain, there's another amino acid and another amino acid that both has sulfhydryl group. So sulfhydryl group will be interacting with one another to form disulfide bridge. Disulfide, you have to take note, it is a strong covalent bond. It cannot be easily broken by increasing temperature. You apply heat, you increase temperature, you cannot easily break down the disulfide bridge. Okay, so and then along the polypeptide chain here you have another amino acid that has carboxyl as the side chain. Here you have another amino acid that has amino as side chain. So carboxyl will be negatively charged, amino will be positively charged. So this side chain will be interacting to form ionic bonds. Okay, so initially the amino acid is straight linear. Due to the side chain that is interacting, it causes the protein to fold into a specific shape. In order for the protein to attain specific shape, in order for the protein to function. Contohnya kan, kalau macam protein yang kita classify dia sebagai globular protein nanti. Okay, globular protein such as enzyme, kan, enzyme kita dah belajar kan, dah belajar kat sekolah since. So the structure of enzyme, it has active sites, kan, active site for substrate to bind. So in order for the enzyme to attain a functional shape, so it has to fall into a specific 3D structure. So this is due to the tertiary structure, due to the interaction between the side chains, either through hydrogen bonds, hydrophobic interaction, and also van der Waals interaction, basophile bridge, and also ionic bonds. So all these involves in the formation of 3D shape of a protein in the tertiary structure. So the next one is quaternary structure. So for this one, the overall conformation of a protein produced by the interaction of two or more polypeptide chain. Keyword here is two or more polypeptide chain. So collagen, we have satu yang biru, satu lagi yang merah, satu lagi yang hijau. 3 chains of polypeptide that are interacting to form collagen. And the other one here given here is hemoglobin. Hemoglobin is globular protein, yang ni globular, yang ini fibrous protein. Okay, fibrous protein. So globular protein, for hemoglobin, it has four polypeptide chain that are interacting to form the hemoglobin structure. So each polypeptide forms a subunit. Yeah. So kita ada alpha subunit, 2 alpha subunit, 2 alpha subunit, and 2 beta subunit, in which each of the subunit has a hem group. Inside the hem group there is iron. That's the ferrum to bind with the oxygen molecule. Okay. So therefore, one hemoglobin molecule can bind with 4 oxygen molecule because it has 4 polypeptide chain that are interacting to form the hemoglobin structure. Itu je lah kan. That is on the based on the level, and then kita tengok next is the structure. So you can classify protein based on its structure. So protein can be globular protein, it can be fibrous protein. Okay, so what's the difference between globular protein? Globular protein are protein that falls into a globular shape. Okay, a globular shape. Globular shape, either the polypeptide chain is coiled or folded. Okay, composed of secondary and also tertiary structure. And then because it is folded into specific shape, again it involves mesti ada tertiary structure. The polypeptide chains are held together by hydrogen bonds, ionic bonds, hydrophobic interaction, disulfide linkage, and van der Waals interaction. And the sequence of amino acid along the chain will be different from one protein to another protein to another protein. The sequence of amino acid is different. Kan? Kita ada enzyme A, kita ada enzyme B, kita ada enzyme C. Each of these different enzymes will have different function, catalyze different types of reaction. So therefore, the sequence of amino acid will be different. Okay, and generally soluble in water. Can so they can dissolve in water because most of our body composed of water. Can your blood composed of mostly of water. So this protein needs to be able to be carried along your blood, so they can be dissolved in the water in your blood. Macam itulah, okay. Soluble in water, and then important function in metabolic reactions. For example, here we have hemoglobin, myoglobin, antibody, and also enzymes. And as for the fibrous protein, they are fibrous. Fibrous, the property of fibrous is that it cannot dissolve in water. Okay, cannot dissolve. So long strands of polypeptide chain composed of primary and secondary structures. Okay. Peptide chains are held by hydrogen bonds because if it's involved secondary structure is hydrogen bond to form helical and also pleated sheets. And then repetitive amino acid sequence. Can so the sequence of amino acid is repetitive. It is the same. Can and then generally insoluble in water. And then important function as structural and supporting protein. For example, keratin, collagen, elastin, fibrin. So fibrin. So these are all the proteins that form structures within an organism. Kan? So keratin in your nails. Kan? So the protein in your nail cannot dissolve. Kan? Kita mandi setiap kali basuh tangan, tak adalah kuku awak larut dalam air. Collagen kan dalam skin. Okay. Keratin in your hair. So all these are proteins that will not be able to dissolve in water. So Fibrous cannot dissolve in water. Globular can dissolve in water. Okay, the next one is based on composition. Simple protein I've mentioned before consists of only amino acids. Okay, so they do not contain any other substances. So example, albumin is egg white, egg white. Globulins, antibody and fibrinogen, globulins. Histone, proteins associated with DNA. Histones are protein where DNA will wrap around that histone protein so that the large molecule of DNA can be packed into the small nucleus within the cell. Okay. Then we have scleroprotein, keratin, collagen in connective tissues. So simple protein consists of only amino acids. And proteins that has other non-protein component, we call it as conjugated protein. Okay, conjugated protein. It has non-protein material, we call it as prosthetic. Contoh tadi apa? Hemoglobin, kita ada hem, dalam tu ada iron, which is function to bind to oxygen, kan? Which is hemoglobin is the pigment in red blood cell, erythrocyte. We have myoglobin, similar to hemoglobin, hemoglobin in blood, but myoglobin is in the muscle. Okay. And then glycoprotein is the combination between protein and carbohydrates. Okay. Glycoprotein is carbohydrates and protein. So basically, where can you find glycoprotein? There are molecule embedded in the plasma membrane. In chapter 3, kita akan tengok lagi detail. And then lipoprotein is the combination between protein and lipid. Okay. Also component of cell membrane. And then nucleoprotein tadi yang apa nama? Nucleoprotein is between protein and also nucleic acids. Okay, so to form chromosomes. Kita ada tadi histone kan? Histone protein is protein. Combine DNA wrap around the histone, banyak dia akan form chromosomes. Okay, Chromosomes consists of protein with DNA wrap around that histones. Okay. And then you can also classify protein based on function. So functions, structural protein, movement, defense, storage, signaling, and also catalyzing reactions. Okay. So structural protein such as, for example, keratin, movement, actin myosin in your muscle, storage in albumin, kan? So storage protein in eggs, the egg white, and then signaling reaction. So this one is enzyme, can be enzyme. Sorry, catalyzing reaction is enzyme, okay? And then signaling is hormones, okay? So I think that's it. Okay, the last part is denaturation of protein. So protein, because it consists of amino acid and the interaction of the protein, it can involve hydrogen bonds or ionic bonds. So these are weak bonds, okay, especially hydrogen bond is they are weak bonds. So you can break the hydrogen bond by heating up the protein. Okay, so when you heat up the protein, you increase the temperature, you break the hydrogen bond. So when the hydrogen bond is broken, the protein loses its original shape and becomes denatured. So therefore, the protein is now will not be functional. It loses its function. So ini kalau apply, one of the factor that causes protein to denature is you increase temperature. The other factor that may cause protein to denature is if you change the pH. Kan? If you change the pH, you change the charge. Kan? You add more hydrogen, for example, you put protein in acidic solution or more basic solution. This will disrupt the ionic bond between the side chain. Kan? So if you change the pH, you may disrupt the ionic bonds between the side chain and causes the protein to lose its original shape. Denaturation can be reversed. Depends. It depends on the situation. So denatured protein sometimes can return to its functional shape when the denaturing agent is removed. If you return back the pH to the original pH, so the protein may attain its original shape, but not all the time. Macam itulah, the chemical and physical aspect of its environment are restored to normal. Okay, so I guess that's at the end of the subtopic protein. Okay, so if you have question, you can ask. Ada soalan ke?
Madam, nak tak?
Ya.
Uh, dia tak akan ada kaitan dengan slide, tapi saya nak tanya esok just for confirmation. Esok untuk lab kelas 10 masuk dulu. So kami tak masuk lab kan, kami buat discussion kan.
You can sit down together to discuss on what you are planning for the mini project. Macam tu lah.
Alright, thank you madam.